Now we will begin Session 7, Reacquisition of Lost Functions for Healthy Longevity. I'd like to introduce our session chair, Dr. KURITA Masakazu, from the University of Tokyo Hospital, who will be guiding us through this session. Our speaker for this session is Dr. Jun Wu from University of Texas Southwestern Medical Center. Dr. KURITA, over to you.
Thank you. Thank you for giving me a chance to talk. Today, we are talking about our respective approaches to generate the functional, to regaining the functions from the developmental embryonic, with the power of the embryonic cells and the tissues. The approaches are completely different from me and Jun, but probably it might be interesting for the people. Please talk. Please listen to that. We are now working on the development of methods to regenerate complex tissues via induction of embryonic developing tissues in situ, which we call tissue embryonization. The core technologies lie in reprogramming and in vivo gene transduction. Generation of iPS cells by Yamanaka is the most popular work in research using reprogramming technologies. In mammals, zygotes generated from ovum and sperm repeatedly divided during early embryogenesis generate complex structures to form embryo and finally born as one independent animal. Especially during embryonic period, the pluripotent stem cells differentiate to various types of embryonic progenitors to generate differentiated cells, which consist of a body. Yamanaka found that transduction of only 4 genes to differentiated cells generates pluripotent cells. On the other hand, similar technologies are utilized between the differentiated cells. It is usually called direct reprogramming. With this, we could change the type of cells by forced gene expressions. I would like to briefly introduce our work today. The primary purpose is the development of innovative solutions for cutaneous ulcers. As I feel, substantial number of patients were suffered from non-healing ulcers such as pressure ulcers and those from peripheral arterial disease when I was a young plastic surgeon. In any kinds of cutaneous ulcers, re-epithelialization can only be attained by migration of surrounding keratinocyte. I mean, epidermis is regenerated only from epidermis, so the healing of large ulcer takes long time. Sometimes the deficiency of the skin could be a cause of the death of the patients. We consider if direct reprogramming of wound-resident mesenchymal cells to epidermal cells in situ, it could cause direct epithelialization from bottom of ulcers, overcome spatial constraints, and which could be innovative therapies. To find out the reprogramming factors which could generate keratinocytes, the epidermal cells from fibroblasts, we started with in vitro experiments. Data from microarray, microRNA, microarray, comparing gene expression of fibroblasts and keratinocyte, and some bioinformatics analysis, 55 transcription factors and 31 microRNAs were picked up as candidate factors. Around 200 combinations were tested over 2 years of period. These are the appearances of starting material and the target. This cell is somewhat cobblestone-like, but not sufficient. When so-called pluripotent genes such as Yamanaka factors are transduced, the shape of the colony is getting closer to ES or iPS cells. Through repeated addition and subtraction, the cells were getting somewhat similar to epithelial cells, and finally obtaining keratinocyte-specific polarized alignment with 28 genes in 2014. It's been around 90 years, and we applied so-called 3-dimensional skin culture to see the behavior of generative cells in air-liquid interface. In 3D culture, 28 PF cells show stratified epithelial structure, which is similar to normal skin, and therefore we named the cells as induced stratified epithelium progenitors, ISEPs. Try to keep the important phenotypes, such as shapes, efficiency of cell production, proliferation capacity of produced cells, and structure in 3D culture. We reduce the factors to 4 factors, which we call DGTM factors. To confirm if in vivo in situ reprogramming of wound-resident cells to ISEPs could generate epidermal tissues in vivo, we introduced isolated skin ulcer model. Suturing synthetic tube on deep fascia on back of mice enables separation of skin ulcer from surrounding skin, and therefore the ulcer in chamber never heals. Epidermis is regenerated only from epidermis. For in vivo gene transduction, we employ the adeno-associated virus vector, since AAV is known for one of the strongest vectors for in vivo gene transduction and known as its safety features. We have tried administration of DGTM expressing AAVs into the isolated skin ulcer. On the day 18 samples, we could confirm stratified epithelial-like structures on and beneath the deep fascia. After 4 weeks, we could confirm visible epidermal structures. On histology, induced epithelium is quite similar to original epidermis located outside the chamber. It was confirmed that AAV-mediated transduction of DGTM factors induced direct reprogramming of wound-resident mesenchymal cells to epithelial cells and generates stratified epithelial tissues similar to original epidermis. To see how much could be expected clinically with the technology, FGF2 and ROCK inhibitors, drugs known to enhance the wound healing, were daily applied to maximize speed of epithelialization from day 4 onward. Enhanced granulation formation was observed after 1 week. Around day 10, the surface of ulcer got rather whitish and scattered small epithelial islands could be confirmed. Wound exudates were reduced around 2 weeks, and most area was closed with epithelial tissues around day 17th. It was confirmed that conceptually, the technology could induce multiple de novo regeneration of epithelial tissues from surface of ulcers, and has potentials to be applied for the skin ulcer treatment of any dimensions. The primary purpose was accomplished, but at the same time, we encountered the fact that epithelial coverage for wound is not sufficiently strong in terms of clinical applications. I mean, I had several occasions to talk with companies and business people, but always judged as the topic is weak to overcome safety concerns toward clinics. For the progression toward clinics, we determined to develop an advanced method to generate skin with higher quality. Regeneration of skin appendage is still a big challenge. We focused that skin appendage is generated de novo in late fetus. We first started to look for reprogramming factors to generate embryonic progenitor-like skin cells and tested them for in vivo generation of skin with appendages. For the screening of reprogramming factors, we used so-called skin reconstitution assay. With embryonic skin cells, we could confirm robust hair reconstitution by cell transplantation to nude mice. No hair was generated with transplantation of adult mouse-derived epithelial and mesenchymal cells. With newborn mouse skin, we could culture epithelial cells and mesenchymal cells separately and obtain substantial hair regeneration on transplantation. To identify the epithelial cell factor, mesenchymal cells are fixed with newborn derived cells and search for epithelial factors which could reprogram mesenchymal cells from adult fat to hair generatable epithelial cells. First, we tried DGTM factors, but it didn't work. After several rounds of testing, we found addition of an embryonic factor, LEF1, confers hair generatable capacity to ICEPs. Next, this DGTM-L ICEPs was fixed as hair generatable epithelial cells and looked for mesenchymal cell factors. It was a substantial story, but after more than 200 testing, in the animal transplanted with 3 transcription factor cells, we found hair-like structures beneath the skin. We thought conferring the embryonic skin niche might help hair outgrowth and try to add the third cells expressing LEF1 and SHH. And finally, confirm the outgrowth of hairs. A stereoscopic observation confirms hair outgrowth from the central portion and mature hair shaft, DP-like structure, and the sebaceous gland were histologically confirmed. With the testing of reduced number of reprogramming factor cells and measuring the number of hair outgrowths, we optimized the factors to DGTML for epithelial cells, FP, and LS factors for mesenchymal cells. Our primary purpose was de novo generation of skin with appendage in ulcer in situ. So, to see if it is possible, again, we employed isolated skin ulcer model and sequentially transduced identified factors using originally developed new AAV capsid. Actual findings of day 0, epithelial factor AAVs are administered. Day 9, mesenchymal AAVs were administered. Day 14, day 21, epithelialization started, and on day 28, we could confirm visible epithelial tissues, and on magnification, only a little, but hair outgrowth was confirmed. On histology, hair follicle and sebaceous gland-like structures were confirmed. Again, I believe the shortest way to push the things forward is the development of the method which could induce the strongest phenotypes. And I realized myself that I prefer 0 to 1 type developmental research than the others. So we are now trying to find factors inducing limb bud like status in amputated limbs of adult mice, which might change a series of limb composing cells to embryonic progenitors for regeneration of limb via tissue embryonization. I'd like to show some data during 2 years of our attacks. Without any additional intervention, amputated forelimb stump of mice heals with cortex bone and thin epithelial integument. In early series of experiments, we found axial elongation of bone stump could be occasionally induced, but it's clear this might be nothing but simple enhancement of normal wound healing. On the other hand, in amphibians with the capacity to regenerate complex tissues even in adult, as mesenchymal tissue mass, which is known as blastema, is formed after the amputation of limb. We consider similar phenomena might be required in mammals and repeated trial and errors. This finding is one that obtained in 2022 spring. Formation of mesenchymal mass at the distal portion of bone stump could be confirmed. Emergence of bone structures from mesenchymal mass could be confirmed in some other factor combinations. This is the finding obtained in last summer, followed up for a prolonged period of 10 weeks. In this animal, all the manipulations such as the introduction was completed within first week, but the prominent changes started after 1 month. Based on the set of genes inducing late tissue outgrowth, which go beyond the cutting plane, somewhat similar phenotypes to other species of animals like lizard or newt. We repeated try and error cycle in terms of types of factors, way of gene transduction and way of surgery. This is the one data obtained early this year. Within 4 weeks after cutting and gene transduction, the generated tip changes to rather flattened shape, When looking from ventral side, some branched structures could be observed on the surface, and on histology, bare bone marrow cells are located just at the side of whitish structures. Until we get to the correct answer, we have to doubt all the procedures on zero-based basis every time, so the progression is not one way forward, but as a general tendency, frequency of emergence of interesting finding is increasing. This is one of the interesting findings of ongoing series of experiments. With some combinations of newly employed factors, the tip of elongating stump tissue seems to start branching several weeks after the completion of gene transduction. One thing clear so far is that with the experimental platform we have developed, we could see the differences between various interventions and resulting regenerations. Still, we are in the position of 0 rather than 1 in terms of 0 to 1 development, but it was always the same, as with skin and with hair. Until the moment I encountered the completely right answer, I couldn't even feel how far is it. And with such an approach like ours, no one still knows if the answer is actually there or not. But in case if the answer would be truly there, I feel we are getting closer day by day. I know this is not easy, but I believe if the answer is there, it would even pave the way towards generation of other organs composed dominantly of mesoderm and ectoderm, such as orbital content and kidney. Nothing is for sure, but I feel we still have to continue fighting for the future. These are the main collaborators supporting the limb regeneration project. Dr. Sakai, a gel scientist, and colleagues already have developed several novel products useful for gene transduction and cell transplantation. Dr. Hojo, helping searching factors, have reported their important findings regarding skeletal development. I'd like all the audiences today to focus on ongoing works of these collaborators, regardless of the direct association with the limb regeneration project, since they are a handful of undoubtedly great researchers I have ever met. And we are fighting for the greatest future of the human race. Thank you for listening. So I'm now touching to the other greatest researcher, Jun.
First of all, I'd like to thank the organizers for inviting me here for this very exciting symposium. Again, my name is Jun Wu. I'm from Department of Molecular Biology from UT Southwestern. My lab is focused on this very unique cell type called pluripotent stem cells. It's unique in the way that it has these two remarkable properties. One of them is called self-renew. They can make copies of themselves over and over again. And the other very important property is pluripotency, is the ability of a single cell to give rise essentially to all the cells, tissues, and organs in our body. And pluripotent stem cells can be derived from a variety of different sources. The most common source is from embryos, such as the blastocyst that's shown in the picture. It's a very simple structure. It contains a ball-like structure, has a cavity and the inner cell mass and outer layer of the trophaloderm. So the green cell showing here is called epiblast. So once you take these cells out in a culture dish and give them a certain combinations of growth factors and chemical inhibitors that manipulate different signaling pathways, we can derive cells called embryonic stem cells from blastocyst. But pluripotent stem cells can also be generated from another source through a process called cellular reprogramming, pioneered by Shinya Yamanaka. Because of this finding, he earned him a Nobel Prize in 2012. And the idea is very simple, is to use 4 transcription factors, OSKM, and forced transfection of these factors into a somatic cells and induce these cells into embryonic stem cell-like cells, oftentimes called induced pluripotent stem cells or iPS cells. So once these cells are stabilized in culture, as I mentioned earlier, it retains these two remarkable properties of self-renew and pluripotency. It gives rise to essentially all the cells and tissues, organs in our body. So my lab is centered on this system, pluripotent stem cell system, and one of the major directions in the lab is to use pluripotent stem cells to engineer tissues and organs so that we can potentially in the future can be applied to humans. Although my lab is not directly study related to aging, but I believe that by having this source of cells, which can potentially give rise to essentially unlimited quantity of tissues or organs, then in the future, maybe we can customize organ replacement, just like if you have an old car, if the engine is breaking down, then you can swap the engine with a new engine. A similar idea apply here. So we mainly taking two different approaches to do that. One of the approaches is to use interspecies chimeras or interspecies process complementation, basically use human pluripotent stem cells, inject them into an animal embryo, deficient in a way that the animals cannot generate a certain tissue, organ. For example, if you knock out a gene called PDX1 in the animal embryos, such as a mice and pig, the mice and pig will not have a pancreas. By using donor pluripotent stem cells from another species, also from humans, potentially we can generate organ or tissues inside the animal species. So this work is pioneered by another great Japanese researcher, Hiro Nakauchi, but I'm not going to talk about that direction today. Today, actually, I'm going to focus on another direction in the lab, is to use pluripotent stem cells to generate early embryo-like structures so that we can use these cells to let them self-organize in 3 dimensions to mimic early development so that we can have a source of functional cell types that potentially can be used for generate tissue and organs in the future. And specifically, I want to talk about a new model that we just published last week and to model this process of gastrulation. So to model early human development or generate early human embryonic like structures, we have to start with pluripotent stem cells. So fortunately or unfortunately, there are many different types of pluripotent stem cells have been reported in the literature. The original report on the human embryonic stem cells actually are deriving the cells that are closer to peri-gastrulation human development. In the case of human, it's about embryonic day 12 to day 13 human epiblast. Even though they were initially derived from the blastocyst, the pre-implantation blastocyst, but these epiblast cells undergo continued differentiation and stabilize as a state corresponding to a post-implantation epiblast. And more recent work by several different groups showing that we can capture a naive pluripotent stem cells, naive in a way that they're more earlier in development, they're corresponding transcriptionally more similar to a pre-implantation epiblast, in this case as embryonic day 6 and 7 human blastocyst. And also there are some cell type reported in the literature are in the middle, for lack of better name, we call it intermediate pluripotent stem cells. So as I mentioned, one of the major goals of my lab is to model early human development. We are very interested in this period of development between week 2 and week 4. The reason being is that this is essentially a black box of developmental period because we have really limited access to early human primary tissues. And not only that, obviously we have accessible experimentation using pluripotent stem cells. We can use pluripotent stem cells to model this development. But more importantly, I think, is if we can model this period of development when all the tissues and organs, progenitors are specified and differentiated initially, then we can provide an unlimited supply of potential embryonic tissues and progenitors for regenerative purposes. But more importantly, by quoting Richard famous saying, is that what I cannot create, I do not understand. So by using proponent stem cells to engineer structures and mimicking early human embryo can help us understand how this developmental program is initially set up early on during human development. And during the week 2 to week 4 of human development, there is a very important process called gastrulation. So gastrulation is important, not only I believe is important, but also by quoting Lewis Wolpat, famous saying is, it is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. Because during this period, the pluripotent epiblast undergo delamination and specifying all 3 primary germ layers, including ectoderm, endoderm, and mesoderm, which essentially give rise to all the different organs and tissues later on in our body. So to model gastrulation, there have been several different models using pluripotent stem cell has been reported in the past, including 2D models such as a 2D gastroid pioneered by Ali Brahmanu's lab, and also Afonso's group uses 3D gastroid model, grow this prime pluripotent stem cell in 3-dimensional so that they can model the anterior-posterior axis formation and also early lineage specification. And also another model from Jianqing Fu's lab called PASE model is post-implantation amnion sac embryos, so model the process of amnion formation, amnion cavity formation, where we believe is the primary germ cells as well as the gastrulations occurring. But all of these models are relying on this particular cell type called prime pluripotent stem cells. As I mentioned earlier, prime pluripotent stem cells are corresponding to this peri-gastrulation epiblast. But what's missing from this model are extra-embryonic cell types, including the placenta cells, trophoblast cells, and also the yolk sac cells. So these cells are not part of the embryo, but they are very important during human development, during pregnancy, gestation, supporting the human embryo development. So as you can imagine, they're providing the growth factors, signaling to help establish the body axis and also signal the onset of gastrulation. So how to model a gastrulation by using extra-embryonic and embryonic cell types? So we test several different starting pluripotent stem cells. We finally decided on this very unique cell type called extended pluripotent stem cells, or in short, EPSCs. So the reason why we choose to use this cell type is because they not only can give rise to embryonic epiblast-like cells, but also they can, under certain conditions, they can be differentiated into hypoblast cells, which is a precursor to give rise to the yolk sac later on. So by developing culture conditions, we can efficiently differentiate initially pluripotent or SOX2 positive cells into 2 different cell types, into SOX2 positive epiblast-like cells, as well as SOX17, which is a marker for extra-embryonic endoderm or definitive endoderm, SOX17. As you can see, after a couple of days differentiation, we can see in this colony, there are 2 different cell types starting to appear. But what's surprising is that this culture condition can also support co-culture of these 2 types of stem cells in the dish for a long time. In this case, at passage 12, we could culture epiblast-like cells and hypoblast-like cells in exactly the same media for a long time. By observing this co-culture, you can't help by observing that they are not random. So in many of the colonies, you observe that the SOX2 positive cells are situated in the center and surrounded by SOX17 positive cells. So these observations suggest that there is a crosstalk between these 2 cell types in a way that they arrange in a specific pattern in 2D cultures. So that gave us an idea. Maybe by growing them in 3-dimensional, maybe we can utilize this co-culture system to help us build a model to model epiblast as well as the yolk sac, one of the extra-embryonic tissue, and study their crosstalk and potentially can model the gastrulation. Why that we choose to use hypoblast is because in mice and many other species studies, we know that the the hypoblasts or actual embryonic endoderm cells are the cell type that actually give rise many different signaling growth factors, inhibitors that establish the anterior posterior axis of the embryo and help initiate the gastrulation. So this is exactly what my very talented postdoc Lee Jun Wu did. So by using a 3D culture conditions, we utilize 4 different steps, develop such a protocol in a way that initially this SOX2 positive cells will undergo lineage segregation, segregate into 2 specific lineages, the hypoblast-like cells and epiblast-like cells. And then we change a condition a little bit to allow these 2 cell types to self-sorting into different compartments. And from that time point, we remove all the factors. We didn't provide any growth factors, signaling molecules, and let these structures self-organize on their own. And after 10 to 11 days, this is what we observed. We started with a very small cluster cells, and on day 2 and day 3, you see these cells grow, structures grow a little bit bigger, and about day 4 or 5, you're starting to see some pattern starting to arise in these structures. And day 6 and 8 is marked by more growth in the epiblast as well as the outer layer of the aggregate. On day 11, you can easily observe there are different structures, organization, configurations has been apparent in these structures. For example, there are 2 cavities. There is one of the cavity seems like it's the amniotic cavity, and the other look like it's a yolk sac. And then in the middle is probably a cell derivative of the epiblast that eventually give rise to all these different tissues and cell types. And we measure the length of the structure, the size of the structures. As you can see from this graph, they continue to grow from the day 1, day 2, up to day 11. And also, it's a relatively high efficient process. So we observe up to 60 to 80% of the efficiency in generating this type of structure at different time points. So this is day 8. We took a scanning electron microscopy image and removed part of the structure, basically the bottom part, which we believe is a primary yolk sac, to expose epiblast compartment. As you can see here, even though we cannot determine which cell types are there, but the structure looks complex and looks very similar to what people deposit online. This is a human embryo. It's at kinase stage 7. So it looks morphologically similar. And more importantly, by the end of the differentiation, a single experiment, we can generate hundreds of these different types of structures. So it's very efficient and very high throughput. So in the next few slides, I'm going to walk you through several different steps of this differentiation. So we use a dual reporter, a reporter SOX2, green color here, and SOX17, red color here. And during day 1 to day 4, as you can see, on day 2, the 2 cell type appear, and day 3 and 4, the 2 different cells starting to sort into different compartments. By day 4, you have these 2 segregated aggregates that are present, and you can see these efficiencies pretty high. Almost every single microvels, you have these 2 different compartments. And by day 5 and 6, you're starting to see cavity formations. And most of the structure are like this. This is from the top view. And from the side view, you can easily observe that there's 2 cavities. There's the amnion-like cavities and yolk sac, primary yolk sac-like cavities in the bottom. And here is the bilaminar disc-like structures, 2-layer structures that are one of the milestones during early human development. And here just another image showing this is a picture of the real human embryo at this stage. And this is our structure, although they're still very different, but roughly they look alike with the 2 cavities and the disc. Here just a little bit more information about this structure at this day, and we use a CDX2 to label the amnion, epithelium, and indeed in this structure it contains an amnion, amnion cavity-like structure, and also at the bottom is a SOX17 encircled a yolk sac, primary yolk sac-like structure. And here just another view from the top, and in the center is amnion cavity, and in the periphery is a primary yolk sac, and SOX17 positive cells is the hypoblast, and SOX2 positive cell organizing the columnar epithelium. And here, using a different marker, you can see this two-layered embryonic disc formed in day 5 and 6. And this is a video just to take you through a different Z-stack of these structures. You can clearly see there's two cavities, one above, one at the bottom, that mimicking amnion-like cavity and the yolk sac-like cavity at the bottom. And on day 6, we also see a primary germ cell-like cells appearing in these structures. We use a reporter line that PRDM, driven by PRDM1 and TFAP2C promoters. So PGC-like cells at this stage co-express these two markers. As you can see, there's several different yellow cells which indicating the appearance of the PGC-like cells in this structure. And more importantly, we observe the migration of the PGCs in day 6 aggregates. And as you can follow these two yellow cells, it's starting to extending the polypodia and seems to migrating somewhere. Although from this video, we cannot see where do they go. It's not responding. Anyway, so by day 8, the structure become even more complex. We're starting to observe underneath the SOX2 positive cells, there's another cluster of SOX17 positive cells, suggesting to us maybe SOX2 positive cells has undergone gastrulation and become different endoderm cells. Here. Yeah. And we look at the axis formation. In this case, we look at the anterior and posterior. We use marker such as SIR1, marking the anterior part of the epiblast. Indeed, at this stage, the epiblast become polarized. And at the posterior end, we're starting to see T-positive cells signaling the gastrulation has started in these structures. And here, just another different looks. We use T signaling the initiation of the gastrulation. You can see these T-positive cells starting to migrate downwards, starting to delaminate and mimicking this gastrulation process. And in day 8 structure, we also can observe a trilaminar disc-like structure formation. So trilaminar disc is another milestone during early human development. During the process of gastrulation, you can see the SOX2 positive cells on top, and then the T positive cells in the middle, and SOX17 positive cells underneath. And by day 9, the structure grows even bigger, as you can easily observe the distinct compartments, mimicking the amnion-like cavities and also yolk sac-like cavities. In the middle is the epiblast. And here, just one of the sections, we show that the yolk sac cavity become bigger, and the amnion cavity also become bigger, and the structure become more complex. And more importantly, by days 10 and 11, we're starting to see early organogenesis is happening. At the anterior part of the structure, we see SOX2 positive cells forming similarly a neural groove or neural plate-like structures. And at the posterior end, by immunostaining with the, by staining with the T and SOX2, we observe a tail bar formation in these structures. And some of the cells co-express T and SOX2, suggesting these cells are neural mesoderm progenitors, which eventually going to give rise to pre-somite mesoderm and somites, and undergoing segmentation give rise to different somites. And here just another image showing that indeed at the anterior part, the neural tube-like structure is starting to appear in these structures. And also these SOX2 positive cells are labeling the neural tube are co-expressing the PAC6, one of the earliest markers of neuroectoderm progenitors. We performed the H&E staining of the day 12 perigastroid. We call this perigastroid because this structure mimicking the perigastrulation development in humans. And compared to a real human embryo at CSA stage, you can see there is a complex tissue formation in these structures, including at the anterior part, the neuroectoderm, and some of the mesoderm cells, and also endoderm-looking cells. Beneath it is yolk sac and amnion sac. So we performed single-cell RNA-seq analysis, analyzing the day 11 perigastroid. We could observe major cell type from all three primary germ layers, endoderm, mesoderm, and ectoderm. And also by comparing existing data set of the human and non-human primate embryos, we found that the perigastruloid cell type are very similar to CS8 stage of the monkey or human development. Here to summarize this study, by starting with extended proponent stem cells, undergoing 4 different steps, we are able to efficiently and robustly generate structure that mimic perigastrulation developing humans. We call them perigastruloid. And here, just the people that highlight in blue are the ones that are contributed to this project. And obviously, inputs from many members of our lab also are important for this study. And with that, I'll be happy to address any questions in the QA section. Thank you.
Thank you for the presentation. We will now proceed to the Q&A session. Before we begin with the audience questions, we have questions from Dr. KURITA to Dr. Wu. Dr. KURITA, please go ahead.
Yes, thank you, Jun, for the beautiful presentation. I'm really impressed with the perfectness of the data, the similarity to the true ones. But as far as I know, I'm not the expert of the fields. I have only a little experience in the pluripotent cells, but as I mature of the field, I feel that the similar types of the approaches, similar types of the papers describing the gastroid, embryoid, those kind of papers, we can see a lot of publications are there. So what's the difference of your ones and the others, and which one is the better?
Yeah, that's a great question. So as you probably heard, there's a number of different studies has been published as well as put on bioRxiv regarding modeling early human development up to gastrulation. And each has their own weakness and strengths. I believe ours are not perfect. Our models also has this limitation. But what's advantage of our model is simple and without transgene, and some of the models using transgene-induced cell fate changes early on. And also our protocol are relatively high efficiency. I believe is the most high efficiency protocol out there. And more importantly, this model developed a little bit further. And I think our model is representing one of the first to model this premenstrual formation, gastrulation, and even a little bit post-gastrulation development into early organogenesis. Yes, there are many different protocols out there, but each has their own strengths and weakness. Yep.
Is there any spaces for you to go farther? You can go advanced.
Yes,
this is the one solution. I don't know. You can go farther.
Yeah, there are 2 things I need to mention here because any type of human embryo modeling we need an ethical approval.
Yes, I know.
And so we work with the Stem Cell Oversight Committee from UT Southwestern. They allow us to culture to day 28. And so, yes, we have a few more days that we can develop this structure to a later stage. But what's limiting factor here is that we observe by day 11 to day 13, these structures starting to collapse. And I think it's become too big, and that maybe the nutrients in the center of the structure are not enough to support the further development. So we're considering to changing the culture conditions as well as to use a roller culture system that developed by Reacquitision. As a matter of fact, we just received their system, so we're testing whether by doing that we can extend the structure to a little bit later stage so that we can see progenitors from different organs.
Yeah, I have a lot of things I want to ask, but probably, is there any questions from the floor?